Hello everyone, I'm Anis Al-Ubaidi, and this is session 37 or lecture 37 within chapter 24 of the Advanced Medical Science Liaison and Medical Monitor Certification course program by CCRPS. The chapter is about conceptual and theoretical monitoring, while the topic for this session is about monitoring of drug framework and drug structure interaction trials. Introduction. The definition will start with which is within the introduction, as drug framework and drug structure interaction trials examine how the molecular framework, core, chemical design, and structure specific atomic arrangement influence the drug's efficacy, safety, and pharmacological activity. These trials are pivotal in understanding structure activity, relationships, SAR, and their relevance to therapeutic outcomes. Key points. Framework assessment and the importance here as the framework, or known as also chemical scaffold, forms the backbone of a drug's design. So the chemical scaffold or the framework forms the backbone of a medication's design or a drug's design. Its evaluation helps determine how well the drug interacts with biological targets and functions within the body. For instance, a robust framework can enhance specificity, reducing off-target effects, while a poorly chosen scaffold might weaken efficacy or increase toxicity. And here is a summarized diagram on unveiling the dimensions of a drug frameworks. As here we have drug framework assessment or drug chemical scaffold assessment on the left, on the right, pharmacodynamic PD and pharmacokinetics PK, target specificity and example of beta-lactam variation. The focus area here as the pharmacodynamics, PD, and pharmacokinetics, PK, trials investigate how core scaffolds influence the drug's action and metabolism, and the target specificity here, framework assessments or chemical scaffold assessments help identify scaffolds that bind only to intended biological targets, improving efficacy while minimizing side effects. Example, an antibiotic trial studied variations in beta-lactam frameworks to combat bacteria resistant to conventional treatments. Researchers discovered that changing the chemical scaffold improved activity against multi-drug resistant strains without increasing toxicity risks. Then we have structure evaluation. And here, the importance as the structure evaluation zooms in on the spatial arrangement of atoms and functional groups within a drug, seeking to optimize receptor binding and functional specificity. Modifications to atomic structures, such as stereochemistry or functional group placement, can profoundly affect a drug's behavior. And here is a summarized diagram on unveiling drug structure evaluation. As on the left, we have structure evaluation. On the right, stereochemistry, functional group substitutions, and example as of ACE inhibitors. The key measures here for structure evaluation. Stereochemistry here, structural studies assess the 3D orientation of atomic bonds to ensure the drug aligns correctly with the target receptor. Functional group substitution, swiping or repositioning functional groups can improve binding affinity, pharmacokinetics, PK, and selectivity. As for example, ACE inhibitors used to manage hypertension illustrate this principle, as here researchers found that relocating the carboxyl group on the molecule improved binding to the angiotensin converting enzyme, increasing efficacy while reducing undesirable interactions with off-target receptors. Data integrity. Importance here as data integrity. Ensuring the accuracy and reliability of molecular data is critical for reproducibility and successful regulatory submission. Errors, bias, and structural data can lead to misinterpretation of results and potential trial failure. And here's a summarized diagram on ensuring data integrity in trials. Left to right, we have validate modeling tools, secure data pipelines, and cross-verify data. Strategies here in data integrity, validating molecular modeling tools, employ computational modeling systems to predict structure-activity relationships, SAR, SAR, and validate experimental results, secure data pipelines, use encrypted and reproducible molecular databases to safeguard sensitive data. Example, a cancer therapy trial relied on digital molecular libraries, cross-verifying experimental data with computational predictions in real time. This approach not only ensured data integrity but also accelerated the trial's progress by identifying inactive molecular analogs early. Tools and techniques. Several advanced tools and techniques are essential for monitoring drug framework and drug structure interaction trials. Structural assays used to evaluate the drug's interactions under clinical trial conditions. These assays provide insights into receptor binding affinity, solubility, and chemical stability. As for example here, surface plasma resonance was employed in in an oncology drug trial to monitor the kinetic interaction between a drug and its protein targets, providing real-time feedback on binding efficiency. While here molecular modeling, computational tools simulate 3D interactions between drugs and biological targets, helping researchers predict receptor affinity and optimize binding energy. And here's an example for molecular modeling in a cardiovascular compound underwent in silico analysis to compare binding energies between different stereoisomers. And here's a summarized diagram for monitoring drug trials, summarizing the monitoring of the drug trials. Structural assays, we have receptor binding affinity, solubility, chemical stability, and for molecular modeling, we have 3D interactions, receptor affinity prediction, binding energy optimization. We have digital libraries. These still within tools and techniques. Here we have digital libraries. These databases house molecular frameworks and SAR data, allowing researchers to cross-reference previous findings and identify patterns. Example, an anti-cancer trial used a library of kinase inhibitor scaffolds to evaluate which framework showed the highest specificity against mutated enzymes. Here we have spectroscopic methods, techniques such as nuclear magnet resonance, NMR, infrared IR spectroscopy, and mass spectrometry validate chemical structures and detect any changes occurring during the trial process. Example, a lipid-lowering agent was tested using NMR, the nuclear magnetic resonance spectroscopy, which confirmed the stability of its CIS trans isomer in, varying conditions. High throughput screening (HTS). HTS here enables the rapid testing of large molecular libraries to detect active compounds and identify the most promising scaffolds or structures. Example, an anti-inflammatory trial used HTS to identify cyclooxygenase inhibitors from a library of over 10,000 scaffold variations. Examples of successful trials. Here we have kinase inhibitor development, framework modifications, improved ATP competitive activity while minimizing off-target effects. And here's a diagram summarizing the overview of pharmaceutical development. And here, all the examples that we have in this lecture coming in this or summarized in this diagram, examples of successful trials. Beta-lactam antibiotic trials optimizations to the lactam core improved efficacy against resistant bacterial strains. RNA interfering agents, structure adjustments, stabilized RNA conjugates for targeted delivery in cardiovascular therapy. Opioid receptor agonist trials, functional group repositioning reduced addiction risk while maintaining analgesic properties. Alzheimer's drug development, monitoring structural changes in amyloid binding molecules yielded improved early stage disease management. Summary tables. Next, we have tables that would summarize what we have covered in this lecture. As here we have table on key components of monitoring, component definition, key actions, example outcomes. Components, we have the framework assessment, which is the chemical scaffolds or scaffold assessment. Structure evaluation, data integrity, patient safety, regulatory compliance. Another table on steps to implement effective monitoring. Step focus, key actions, example outcomes. Steps we have protocol development, real-time data collection, regulatory compliance, risk management, and data reporting. Table on tools and techniques for monitoring. We have the tool technique, purpose, key features, advantage, and then example implementation. Tools or techniques, we have structural assays, molecular modeling, digital libraries, spectroscopy, for example, IR, which is the infrared, and NMR nuclear molecular resonance. And then we have the high throughput screening. And NMR here is exactly the nuclear magnetic resonance, nuclear magnetic resonance, and IR infrared. Here we have a table on examples of successful trials. Trial context, focus, key challenge, solution applied, outcome achieved. And trial context, we have kinase inhibitor design, beta-lactam antibiotics, RNA agent trials, opioid agonist studies, Alzheimer's therapies. Application examples, scenario focus, drug examples, approach taken, and impact on outcomes. As scenarios here, we have cardiovascular therapy, SAR, antibiotic scaffold testing, neuroprotective agents, anti-inflammatory trials, rare oncology treatments. Application table, scenario for each scenario, key principle applied, key actions, timeline, what to watch out for. Scenarios here we have monitoring beta-lactam frameworks and antibiotic trials, examining stereochemistry effects on ACE inhibitors, refining kinase inhibitor frameworks for oncological therapies, validating chemical integrity with nuclear magnetic resonance NMR for lipid-lowering agents, screening anti-inflammatory drug scaffolds, with high throughput techniques, cross-validating SAR data and kinase inhibitors using digital libraries, implementing predictive molecular docking in psychotropic drug design, managing stereoisomer reactions in cardiovascular drug trials, adjusting degradation-prone structural frameworks in anti-malarial trials, developing SAR-based substitution rules for opioid receptor analgesics, conducting spectroscopic evaluations of Alzheimer's amyloid-binding molecules, screening paralipid RNA, conjugate frameworks in RNA interference drug trials, reporting SAR-linked statistical trends for Alzheimer's related biomolecular maps. Next, we will revise what we have covered through this lecture through some MCQs or multiple choice questions. Question 1. What is the focus of drug framework interaction trials? A, to study market potential for new pharmaceutical designs. B, to analyze the role of a drug's core chemical scaffold framework in biological activity. C, to evaluate environmental conditions affecting drug stability. D, to compare patent protection protocols across regions. So the answer is B, as to analyze the role of a drug's core chemical scaffold in biological activity. As a framework is the chemical scaffold. As a drug framework interaction, trials analyze how the core scaffold chemical design relates to pharmacodynamics, PD, and pharmacokinetics, PK, optimizing efficacy and minimizing side effects. Question 2. Why is structure evaluation important in trials? A, it predicts manufacturing costs for large-scale production. B, it quantifies the stereochemistry and functional group placement for receptor binding efficacy. C, it focuses solely on toxicity assessments via random sampling. Or D, it ensures marketing strategies align with biochemical properties. So it is about the structure. So B would be the most correct answer in between the four choices, as it quantifies the stereochemistry and functional group placement for receptor binding efficacy. As structure evaluation examines how specific atomic arrangements like stereochemistry and substitutions enhance target binding and selectivity, crucial for improving therapeutic outcomes. Question 3. What tool is most suited for validating chemical structures in trials? Is it A, high resolution cameras for physical documentation? B, spectroscopic methods such as NMR, nuclear magnetic resonance, or mass spectrometry? C, patients' feedback journals, or D, standard pre-filled trial templates? So it is B, the spectroscopic methods such as NMR or mass spectrometry, as spectroscopic methods including NMR and mass spectrometry are essential for confirming chemical integrity and structural arrangements during drug trials. Question 4. How does molecular modeling contribute to drug structure trials? A. By tracking drug cells performance. B. By simulating 3D receptor ligand interactions to predict binding performance. C. By focusing on the economic feasibility of chemical synthesis, or D, by generating random combinations of chemical structures without validation? And the answer here is B. The answer is B. By simulating 3D receptor ligand interactions to predict binding performance. As during the lecture, when we went through the molecular modeling, 3D came as an example, which is B here, by simulating 3D receptor ligand interactions to predict binding performance. As molecular modeling enables researchers to visualize and predict receptor binding interactions, optimizing drug efficacy through precise structural adjustments. Question 5. What challenge is addressed by integrating digital molecular libraries in monitoring? Is it A, difficulty keeping trial costs low? B, lack of diverse demographic representation in early stage trials? C, insufficiency in comparing scaffold models across trials? Or D, limitations in investigator training for molecular testing? It is C, the answer, as inefficiency in comparing scaffold models across trials, as digital molecular libraries provide a centralized database for storing and comparing scaffold models, enabling efficient cross-trial data analysis and optimization of SAR studies. And here we come to the end of this lecture. Thank you very much and goodbye.